Fighting Blindness Canada's Viewpoint Education Series shares the latest in vision research, provides opportunities for you to ask your questions, and connect with other people who are living with a blinding eye disease. The session you're about to watch is a recording. To learn more about the research we fund and upcoming programs and events, please visit our website at fightingblindness.ca. Viewpoint is proudly presented by our Platinum partner Bayer and supported by our Gold partner Roche and Silver partners AbbVie, Astellas, Beacon Therapeutics, Johnson & Johnson, and Specsavers. Thank you for watching. We hope you enjoyed this session and share your thoughts with us in the comment section below.
Welcome back. I hope you had a nice short break. We're going to be doing our panel discussion now, which we're calling From Research to Reality, Conversations about Clinical Trials. During the session, we're going to explore how new therapies for vision loss move from the research lab to patients and meet some of the people behind that journey. So today we're going to be joined by Dr. Gamm, who you just heard from in the beginning. We also have on the farthest from me there is Dr. Robert Koenekoop, who is a clinician scientist at McGill University, who leads several gene therapy and drug clinical trials. And then next to him is Dr. Shaheer Aboobaker, who's a retina specialist and principal investigator on more than 20 clinical trials. And right next to me, we have Bernie Zinck, who's a musician, advocate, and trial participant who's going to share a little bit about the patient perspective. And then we're going to take some more questions. I know there was lots of questions in the first session, so we'll try to leave a bit more time for that. So let's start with clinical practice and how some of the research that we've heard about and other research connects directly to patients. So I'd like to start with Dr. Aboobaker, if I can. What motivates people to take part in your clinical trials, and what sort of concerns or barriers do you hear most often in clinic?
Yeah, thanks so much for inviting me here today, and thanks to Fighting Blindness Canada for everything they do for us and to support us and our patients. And Dr. Gamm gave a great talk that I really enjoyed. The question regarding the trials. So I think there's a lot of sort of patients who are obviously struggling with vision impairment and visual disability, and there's also a lot of desperation for something that we've been waiting a long time for. So Dr. Gamm expertly showed the timeline and how long we've been waiting. And in retina, and I practice sort of adult retina, my pediatric colleagues here do a much more important work than I do. But we've had sort of nothing from macular degeneration for the longest time, and then 12 excuse me, I'm ODing on lozenges to keep my throat going, so I apologize. But then we had these anti-VEGFs that came out and changed the field. And there has been nothing since, and now we're really getting towards a golden age of new things coming. So I think the patients are interested in new therapies. I think they want to know what's going on. They want to be involved in the clinical trials. I think the barriers that we find is, a lot of it is distance and travel. And Dr. Gamm mentioned that, yes, we're able to support you with travel, but we have 4 sites across the GTA. And so my patients that I see out in Oshawa, they can't come to our research site, which is at Don Mills and Lawrence. It's just too difficult for them. So that's one part. I think the other part is education. And I was going to speak about the phases, and I'm glad that Dr. Gamm has handled that already. But it's important to know what phase of trial you're going in for. Not every clinical trial is the same. A phase 4 clinical trial is very, very different from a phase 1. Those are important for us to understand the power of our therapeutics that have now come to market. And so that's going to have much less risk than something a little earlier in the pipeline.
And Dr. Koenekoop, I know you've been involved in trials for many years. So what sort of lessons from previous trials are sort of shaping how new trials are being designed and implemented?
That's a great question. First of all, thanks for inviting me to be here. Great talk by Dr. Gamm. Thank you. And Fighting Blindness Canada for all their support and organizing educational sessions like this, but also supporting amazing research in Canada and beyond. Obviously, we're learning from each trial. And like Dr. Gamm said, there's phase 1, 2, and 3. And the number of patients per phase are different depending on the frequency of the disease. But what we're learning is that we're getting away, and we're teaching the FDA to get away from the measurements for glaucoma and diabetic retinopathy. I think that's one of the major things that we've been able to accomplish in addition to showing safety and efficacy of gene therapy and stem cells and medications, we're finally, with mobility course, with the easy line, which is the number of photoreceptors that you can measure with an imaging that is available in most ophthalmology offices. We have these amazing new tools to measure vision. And the FDA is coming along with us and approving them. A mobility course is I think the most exciting, maybe most exciting new tool where we have a big room with obstacles and different lighting conditions, and we video the patients before the medication is given, before the drug, before the gene therapy, before the stem cells. And then we do the treatment, and then we do the video and the mobility course again. So much I can do. And we don't interpret the data. We send it to a video reading center, and they determine how many mistakes were made before the drug and after the drug. How fast did the patient move through the course? So this is very exciting because it's very, very new. It combines a lot of different features of vision, including peripheral vision, central vision, contrast, you name it. It's all combined in how a patient moves through the mobility course. Very quickly, we also have pupillometry, and we have very exciting new perimetry where we know exactly in the retina where we're testing the sensitivity of the retina before and after the drug, so we can see in an area, for instance, that we are targeting with the gene therapy or stem cell therapy, we know where that area is, and we can measure the sensitivity of that location before and after. So yeah, we're learning a lot. Also safety features. We're learning to be very strict about the entry criteria. And this is kind of an interesting topic because we as physicians who are in this field, we want to help all the patients, right? But we can't help all the patients right away. We want to hit a home run, right, with the phase 1, 2, and 3. And then, because we're only doing this for one reason, to get these medications to the market, to you.
And it seems to me, to Dr. Koenekoop, that some of the new measures you're talking about are more about sort of people's practical experience as well, right? So it's just not numbers on an eye chart. It's about how they're navigating and experiencing the world after the treatment.
Very important. So those are called patient-reported outcomes. And those are questionnaires that are given before the medication, before the drug, and after the drug. And you can analyze the differences in the answers. So you have the patient perspective is crucial, because if we say that the perimetry got better and the patient says, but I can't see better, that's super important, right? So the patient-reported outcomes, and that's the exciting thing. When things start to correlate, patient says, I can see red for the first time in 10 years, and then the red test for the red sensitivity is better. Those correlations are absolutely beautiful and important. So we do all those things all at the same time.
Thank you. And so, Bernie, I'd like to turn to you now. So you've actually participated in a clinical trial. Can you share what that experience was like for you from sort of first hearing about it through being part of that process?
Sure. I'll start with how I sort of found out about some of these trials. And I think it's important that everybody in the room knows Fighting Blindness Canada has a patient registry. So if you're not on the patient registry, that's a good place to start, because these guys will be looking at these patient registries for possible candidates for trials. The second thing I did was there's a great website called clinicaltrials.gov, which has all of the trials that are coming up and underway, and also some that have been completed already. And that is a great place to basically contact the researchers. That's what I did. And so I've been screened twice for a treatment trial. I did not get in, 2 different companies, because the inclusion criteria is quite strict. They're looking for, quote unquote, their best athletes, if you will. So they want the best candidates to get the best results. But I did I was in the Janssen Mirror GTX natural history trial. So I participated in that. That was 3 visits, 6 months apart. And I did the maze that Dr. Koenekoop spoke about. I did all of the tests. And I'm from rural Nova Scotia, so, you know, coming to Toronto at Sick Kids with Dr. Heon here in Toronto, that's where we did a lot of the testing. They have so much better gear, like not to you know, but seriously, like there's a lot of great gear they have here for testing our eyes. So I was able to go through all that stuff. If I had to say anything about my experience, it was quite awesome actually. Everyone was very professional. You know, I had nothing to worry about. You know, sometimes you think trial it could be a little scary, but my experience was it was awesome. They also, you know, it's true, they pay for everything. You know, our airfare, our hotels, food, all included. Like Dr. Gamm said, if you see something where they want your money, just run away as fast as you can. So yeah, all in all, I had a great experience with my clinical trial.
And so, Bernie, you mentioned that the trial that you took part in was a natural history trial versus sort of a treatment trial. Could I ask maybe Dr. Koenekoop or Dr. Aboobaker to talk about what the difference is and why natural history studies are actually very important? You can battle it out.
For each major disease type, we want to know per year or per 2 years how certain aspects of vision decline. So we have that information now for some major types of RP, retinitis pigmentosa. So we want to know, to help us with the drug study or with the molecule or the treatment, we want to know what happens without the treatment. And so we want to know how many millimeters the photoreceptor line decreases per year with the imaging. We want to know the retinal sensitivity, how it changes per year. And we want to do that for many patients so that we have a nice overview and a variability of how that particular aspect of vision declines. And then after a few years, we have that information, for instance, for a gene called USH2A, which is a major gene, so that those slopes that we then create, that we use them in the treatment trial. And that is very impressive to us and the patients, but also to the FDA. So they see that for this particular gene, the visual field goes down, the vision itself goes down, the contrast sensitivity goes down, et cetera, at a certain rate. And then we give the treatment. And then the decline stops. And hopefully the curve goes up. But just the fact that it stops is already a signal, a therapeutic signal.
Yeah, I'll just add in, these are super important studies because, and I'll speak to the sort of the dry AMD and geographic atrophy for, you know, anyone living with this, you know that this will eventually cause loss of central vision. And the problem is that when you are entering a new therapeutic area where there's no therapies, the bar is not just low, the bar doesn't exist. And so for regulatory authorities, the question is how do you compare that this drug is actually working? And so as Dr. Koenekoop mentioned, the natural history study sets that bar against which the new product is tested against.
Thank you. So Dr. Gamm, I'd like to go back to you. So your keynote was fantastic, and one thing that you talked about was sort of setting people's expectations, right? So the work that you're doing sort of sits at that intersection of science and hope. So how do you keep patients' experiences and perspectives in mind as you develop therapies?
I think as a clinician scientist, that's one of the things that I think you have a kind of insight into because you know how vulnerable folks are at. I mean, and can I just make a segue here? You can tell a pediatric ophthalmologist by our socks, right? So I've got like dozens of socks. I almost wore my Star Wars socks today. And I thought, oh, that's not going to be professional. And Dr. Koenekoop, he's got penguins. So yeah. So anyway. I won't make that mistake twice, but I think, again, when somebody comes in and it's palpable in the air, and the family, they know something's up with their child's vision, but they're hoping that it's a pair of glasses or something like that, and you look in there or you do some testing and you know that they've got a condition that you can't treat right now. It's going to be inexorable in terms of its decline. And you know in your heart of hearts that this child's going to do amazing things and the family's going to be good. But it's tough right there. And so witnessing that kind of vulnerability, I think, immediately ingrains in you the sense that you have to be honest, you have to have integrity, you have to treat them like you would treat your own family. And that means education. And that's where Fighting Blindness Canada, other foundations, really have that ability to synthesize information that's out there and provide good, honest information, and that's also our role too. And you should understand too that there are plenty of practitioners out there who mean well, but they're reading things on the internet too, or they may not have the same background or deep knowledge of the different therapeutics. So I would say, you really do want to reach out to FBC. You want to reach out to academic centers that have ocular geneticists who really work in these areas, because most folks aren't going to intentionally lead you astray, but they may very well do that inadvertently too. So.
Hearing Dr. Gamm talk about education is music to my ears as the Senior Manager of Education at Fighting Blindness Canada. And so I will do a slight shameless plug. Bernie mentioned the patient registry. Absolutely encourage you to look into that if you are living with an IRD. But we also have our health information line where we can help you find resources. That clinicaltrials.gov website is very complicated and confusing, and we can help make some sense of that. So if that's something you're curious about, please let us know. So Bernie, just kind of off of what Dr. Gamm said, what sort of advice would you give someone who might be considering joining a clinical trial for the first time? Sure.
You have to think about not maybe just about yourself, but your family, how much travel is going to be involved, how much time is involved. I remember going down to Boston and getting screened for the Mirror GTX trial, and it was presented with the sticker shock, which was essentially, every 2 weeks for a while, I had to kind of travel to Boston from Nova Scotia, which is a lot, So, and then after that, it kind of tapers off. You get gradually longer durations between visits, but you have to think about how you're going to navigate that. Myself, I need assistance in airports and things like that. So my wife, it's her birthday today, by the way. She's responsible for getting me to all these things. So if it wasn't for her, it'd be hard to do any of this stuff. So you have to think about your support system and how that's all going to work.
So I want to get to audience questions because I know we didn't have a lot of time in our first session. So I would like to ask our three doctors this question. So looking ahead 5 to 10 years, what is giving you the most hope for the future of vision loss research? And maybe I'll start with Dr. Koenekoop.
Yeah, I think about that all the time. I do. I was going to mention in this meeting that the last few weeks, I'll let the other two MDs also talk about this, but the last few weeks, we've had 2 or 3 major breakthroughs in treatments for retinitis pigmentosa, macular degeneration, and vision loss. And I'll just quickly mention them, and then maybe we can discuss it later. But there was a paper that came out last week from the New England Journal where a tiny chip was placed under the bipolar layer, replacing the photoreceptors. And the patients, on average, gained 4 or 5 lines on the vision chart. And so that could be very exciting for a lot of people, not just for macular degeneration, but also retinitis pigmentosa often has macular changes and degeneration as well, Stargardt's, et cetera. But I think when I think about the history of medicine and penicillin, how it came about, and then insulin. And the big difference for me with inherited retinal degenerations is that we have 5 or 6 things that are working in clinical trials. Some in phase 1, some in phase 2, many already in phase 3. So I'm extremely lucky to be in this field, and I'm very not just excited, but I'm very hopeful because we have so many different options, including what Dr. Gamm spoke about, to transplant photoreceptors. I'll just take another moment, and I think one other treatment that we haven't spoken about yet, which is really, really exciting because of its elegance, is the antisense oligonucleotides. So for 2 different diseases, CEP290 is a form of congenital blindness, and USH2A is more of a typical retinitis pigmentosa type. We have technology now in clinical trials. One is in phase 3 and one is in phase 2, where we inject into the vitreous an RNA molecule specifically to remove a mutation and leave the gene intact, or remove a segment of DNA where a lot of mutations take place without harming the gene. So gene therapy means you're replacing the whole gene. We've spoken about that, and you know about this. But gene editing, where we remove the error, we remove the mutation, leaves the gene alone and it starts to work again. So we have some very beautiful early data where patients improve in the mobility course, their photoreceptor line gets bigger, etc. So this is called editing. I think the answer to the question is we're very excited about the 5-year future.
Yeah, I think if we take a step back and look back maybe 20 years and sort of look at where we are now. I mean, the things we're talking about today, 20 years ago, was science fiction. You know, we're talking about taking a deactivated viral vector, putting in some self-replicating DNA or RNA, and plug it into the eye and having this become a drug factory. So, you know, all of these things are just, there's so much going on. You see it at the meetings, there's so many companies, there's so many resources, there's so many topics, there's new therapeutic areas where we've never ventured before. And so I think there has never been a better time for us to be in retina, but also hopefully for the patients to be able to deliver on some of these promises. The things I'm most excited about in my particular practice is the stem cells, and specifically in what we do for geographic atrophy. So, you know, you should be able to, again, according to what Dr. Gamm sort of showed about pluripotent cells, replace some of those damaged or dead RPE and photoreceptors. And some of the phase 1 studies, there's 2 that I'm aware of that also, like Dr. Koenekoop said, improve vision by 3 lines. And that could be life-changing for somebody to go from not being able to take care of themselves or orientate and walk around to be able to not bump into objects. You know, that could be life-changing. So there's a lot of interesting, you know, optogenetics was mentioned as well. You know, that's changing the actual structure of the retina. There's cells that, you know, give the retina its structure. Think of the pillars here or the drywall, and then you turn that potentially into a low-resolution television, for example. So there's a lot to be excited about.
Dr. Gamm?
So I'm gonna put on, so I always kind of bias myself a little more towards the basic science or the PhD end of what I do. So what I'm most excited about is all the things that were mentioned here too, but our understanding of the biology underneath what's caused this. I mean, there's hundreds of different genetic mutations or genes that are affected that cause blindness. And within those individual genes, there could be hundreds of different mutations that cause that gene to function inappropriately. And there's an explosion of different approaches, and we're able to customize now. It's not just what gene you have, but what type of mutation. So there are particular types of mutations. Let's give you one example called a nonsense mutation, where it's a mutation that causes, when you make a protein, it prematurely tells you you're done. And you're not done. You're supposed to make a protein that's this long, but it tells you to stop right here. And that accounts for about 20+% of all diseases in your entire body. So instead of just making a therapeutic that can fix that one mutation out of 200 in that one gene out of 200 that causes an already rare disease, can you make something that can read through or bypass these broad categories of mutations, that's more also attractive to investors and for companies. And so you have these Venn diagrams now of genetics where you're replacing a gene, or you're doing base editing, or you're doing ASOs, or you're doing all these different things. And so knowing where your mutation is and what type of mutation it is really critical, and we're learning that. And so that just gives us more shots on goal. And then in addition to that, we're understanding more and more why it is that those mutations end up causing your cells to die or become dysfunctional. That allows us to build what sounds mundane, but model systems in a dish that can give us better and better readouts about whether the therapeutic you're testing actually has a decent chance of helping a human. So that de-risks the whole process. And so that's all done, and it's all done prior to what goes into your eye. And that's so important because you got two, probably one to use in a clinical trial, and you really want to put your best eye forward for all these different things. And so being able to provide that information, get really customized about the model systems that we're using, that's what excites me the most.
Thank you. And Bernie, last word to you. What does hope for the future look like for you, based on what our conversation today?
I think it's looking pretty good, honestly. When I was 16, I was diagnosed with RP, and this was back in the 90s, so I'm dating myself a little bit. But basically, we didn't really know a lot about RP. So and so your doctor has to tell you, sorry, there's no treatments, there's no cures. You're eventually going to go blind, And so that's what I was told as a 16-year-old, right? And here we are, 35 years later, or so, almost 35 years later, and now there's optogenetics, there's bionic eyes, there's stem cells, there's gene therapies, there's probably like 40, I've read somewhere it's 40 or something clinical trials right now. This is crazy, right, compared to what we were told when I was a kid. So, and this is the beauty of everything that's going on right now is that if there's a 16-year-old kid out there that gets diagnosed tomorrow, the chances are they're going to know a lot more. And in my case, if they have the same gene as me, they'll still have a little vision when they're almost 50, and that's probably great news to hear, honestly, versus going blind. So.
Thank you. Okay, we're going to open it. Yeah, thank you. We're going to open it up to questions now. We're going to give a couple of mics out. So if you have a question, if you want to wave your hand, someone will come over to you.
And why don't I just start from one from the
we'll start from the live stream. Sure.
Yeah, and if anybody else has questions for the live stream, please pop them into the Q&A. I think this, maybe I'll start with Dr. Aboobaker. A question about, since macular degeneration is generally associated with older people, is age a major determinant to participate in these trials, or do younger folk get in easier? And I know it's across the board, but I'll start with Dr. Aboobaker for that one.
Yeah, so that's a great question. And the answer is yes and no. So it really depends on the trial that you're signing up for. As we've discussed and Dr. Gamm pointed out, there are inclusion and exclusion criteria, and sometimes one of those is age. So the recent gene therapy trial that we did for a product called Surovec, which is a subretinal gene therapy for wet macular degeneration, they did have a cutoff of 90 years. So it really is trial dependent, and there's specific reasons for that they work towards. Remember, these trials are really about proving the drug to the regulatory authorities once they hit the market. That doesn't mean that if you're above 90, you can't get that therapy.
I'm thinking about some less drastic cures. Can you hear me? Mm-hmm. Occasionally when I listen to some American blogs, I get advertisements that break in. You're going to turn the volume up. So I get some advertisements breaking in, claiming to cure various elements, and they usually are given by some older men like myself who can no longer clearly see their grandchildren and do a number of things. But remember, when they were in North Borneo, that some of the local indigenous peoples could see clearly because they took a certain herb. So we would take the herb back to the United States, and in a lab they would mix that with lutein and vitamin D and maybe even a little Viagra or something, I don't know what it is. And they'd mix it into a pill form and do clinical trials, and eventually I think got FDA approval. I'm talking about one in particular, Retina Clear. So this one guy would give it out, would try it out himself, and he recovered his 20/20, gave it out to his church group, and they were all cleared up. And in the next scene, you could see them dancing around a maypole in merriment or something like that. So I'm wondering what you think. And his point was, why give millions to the eye industry, rich millionaires like yourselves?
Give it to me.
Yeah. And for a limited time offer, you too can order some of these pills and see clearly. Now I'm wondering what you, because obviously the optical industry is a target. I'm wondering what you think of these guys. Are they charlatans, or do these things work? What do you have any strong opinion?
Are you serious with this?
Yes, I am. Yes, I am serious.
I mean, I think you answered your own question there a few times in there, but I think the answer is no. They're right. It's for profit, right? So small profit. No, not small profit. It's small for you, and they're making mostly on shipping and handling, by the way. So they're only making $20 off you, but they're banking on there being 50 million people who just go, hey, they may have cataracts, they're just broadly, right? So it's millions and millions of dollars, right? Not per treatment, because you're not going to really be able to do that in between your whatever newscast or blog you're looking at. But I think stepping back a little bit, I hear this a lot. It used to be, and Dr. Koenekoop probably remembers this as well too, but right in the beginning of stem cells, there was this deal with optic nerve hypoplasia, right? So babies are born with optic nerve, their optic nerve is small, it's congenital, it's genetic. They tend to, when they're born, they act completely blind. They have, their eyes move all around, they don't focus on things. If you let them go a little bit longer, a couple of years, they tend to develop some vision, and they'll stabilize, and they'll get a little better. And so once stem cells got on the scene, there was a Chinese company that said, and it was brokered through a nurse practitioner in Chicago, so it's close to where I am. And if you had optic nerve hypoplasia, you say, oh, come to China, we're going to give you a stem cell therapy, and you'll see better. And so it was brokered through through Chicago. They'd send them over there. It was $50,000. The child would get something taken out of their bone marrow, stuck into their eye or near their eye. They'd come back, and a year or two later, they'd see a little bit better. Right? It was the natural history of the disease. And then guess what? That $50,000 was raised through Tootsie Roll sales in their local community. So they come back, and they're in their local newspaper saying, oh, Johnny sees so much better now. Oh, thank you so much for all that you do. And then the company puts those little small town newspaper articles on their website. And then it goes more, and you can see it's sinister. And if you had the audacity to say, my Johnny isn't seeing any better, they'd say, well, for $10,000, come back, we'll put you in a hyperbaric oxygen chamber because they're not working well, they need more oxygen, right? So, snake oil has been around forever, and it's around now. And perfectly intelligent people who would never, ever, take a cardboard box and say, yeah, this is a great new car, this is good, I'll do it. But when it's dealing with your vision or your loved ones, we lower our bar. So the other thing to remember is, if there was a cure for blindness, do you think you'd have to go to Ecuador to find it? Do you think there might be a company somewhere that would want to make a gazillion dollars off of that, right? It just doesn't make sense, right? There's no hidden cure somewhere that people are suppressing, right? So again, it's mostly common sense, but it's important to be able to step back from your own condition or your condition of your loved one where you're so desperately want to do something and not believe those folks. And the easiest way to do that is dollars. If you have to pay for it, it doesn't work.
So your first step is an ophthalmologist?
Depends on the ophthalmologist. right? Where's their bread being buttered? So, I mean, but yes, I mean, your first step is to get good information and from more than one source.
I'll just add quickly, and this may not age well because we may have sort of better advancements in the future. But if you look at all the therapies we're talking about today, it's subretinal surgery, right? Subretinal gene or cell therapy. You're looking at intravitreal injections and then obviously topical drops. We haven't quite yet cracked the nut on tablets to fix problems. And that goes down to the basic biology that it goes through the liver and it doesn't get high enough to the eye. We may figure that out, but I would say just be wary. It's not a catch-all. Don't take this as if it's a tablet it doesn't work. We may find that in the future, but I would say just question it if it's a tablet at this point for the eye.
If I could say one more thing too, because you said it was FDA approved. It is not. If you are labeled in the U.S. as a nutraceutical, if you're basically a vitamin, or if you are able to label, FDA doesn't regulate you at all, right? So that I can guarantee you that is not an FDA regulated. And so I can sell you my grass clippings, call it a nutraceutical, and say it'll cure your cancer, right? You can sue me civilly, but it's not a criminal issue. So realize that as well.
Thank you. I was wondering about Korea South Korea is targeting the PROX1 protein. I wondered if you knew about that or if we had anything similar, that type of research going around here. And also the optogenetics, is it coming up for FDA approval in the next year, I believe, in the USA? Did you hear anything about that?
I know the FOX1 genes are important in development, but no, I think, so I don't know anything specifically about that. I also do want to say that, there is good science being done at universities around the world, in Europe and in Korea and Japan and places like that. It's not like there's only North America is the only place to look for these trials. So there is good science being there. It's just harder to assess them when they're not in your own backyard. And then in terms of optogenetics, there's been a lot of promise that's gone into that. And so usually when I give a talk about cell-based therapies, you can either replace what you've lost or bypass it, right? So just say, well, the heck with photoreceptors. Let's just make the things that are still there work differently. And that's a great way to go, and especially for patients who have no vision whatsoever. And so, understanding the landscape, understanding where your best shot is important. But the beauty of it is that there are lots of them, which I think I started out by saying, which is a blessing and a curse, right? If you got one hammer, everyone wants the hammer. Now you got 200 hammers. How do you educate yourself to be able to know which one's best for you?
Do you really think that, when I first came, my first time, about 20-some years ago, and they thought it was very exciting, new things had just started happening. So of course, they thought within 5 years there would be a treatment that would be, and then when I came back every year, right? But then they said 10 years. And then now I heard you say 5 years. You think, is that something that is reasonable to think that within 5 years we'd have some treatments for most people?
Yeah, so obviously we understand the need for a timeline and the need for hope. And the last thing we want to do is quash hope. I mean, I think we all work in the field of hope by being in clinical trials and basic science. The problem with this is that when you work in drug development, the vast majority of products fail somewhere along the line. And so we get excited and we discuss the early stage clinical trials and we think this is going somewhere. And inevitably, you and I get disappointed when we find that there's a really bad outcome and somebody goes blind. So it's a tough one because we want to keep you motivated. We want to keep the hope alive. But there are challenges along the way that just are part and parcel of drug development.
Thank you very much for your presentation. I'm speaking on behalf of my friend, Mr. Laku. He had a gene tested in Finland, and he his diagnosis is OSH2A associated retinal dystrophy, retinitis pigmentosa, autosomal recessive. Now, apparently, this, according to your presentation, he would come under gene therapy rather than cell therapy, if I'm correct. And if it's the gene therapy, does the cell therapy become out of question? Another thing I would like to point out that we did some basic research with AI, and we found that there was a therapeutic medication by PTC Therapeutics called Translearner. And apparently, when we contacted PTC Therapeutics, they said they would not inform any kind of such treatment to any public, which makes sense because they don't want to be liable if something goes wrong or what. I'm not sure if you have any knowledge about what I'm speaking about, Translearner. Actually, what we learned is that they had a medication approved by FDA in 2021, but FDA removed the approval a year later, not because it had any side effects, but why they removed it because it didn't show much progress.
So I'm going to let Dr. Koenekoop answer that because we're running a bit low on time.
Perfect. So USH2A is the most common gene mutated in retinitis pigmentosa. And many of the mutations in USH2A are in one particular area called exon 13. And some brilliant scientists in Nijmegen, the Netherlands, figured out that if you remove exon 13 with an ASO, with an editing machinery that I spoke about before, then you remove the mutation, but you don't harm the protein. And that's what we call RNA-based gene editing, where we remove the mutation or a tiny region. And this is now happening in a clinical trial, phase 2 in Canada. So if your friend has the mutations in USH2A and in exon 13, then it would be very important to either come to Montreal or to Toronto, where we are doing these studies. And we're very excited about the preliminary results. If the mutation is outside of exon 13, then we have to wait a bit longer. Why? Because USH2A is not only the most common gene for RP, it's also very large. So it doesn't fit into the viral vectors that we are using to deliver the genes under the retina. PTC, I'm not familiar with. I hope that is beginning to answer some of your questions.
Okay, we have time for one more question. Ali?
Hi, my name is Ali, and I have RP. Before I ask a question, I quickly want to share that I'm wearing socks with subatomic particles on it. And if this wasn't a clue, I'm a nuclear physicist by training. Okay, so my question was, this was asked earlier about the age cutoff. I wanted to understand a couple of things. One, you had mentioned earlier in the keynote that you also look for other conditions like heart disease or something like that. So can you explain a bit more on what other conditions can be cut off for clinical trials? And the other thing is there like a point of no return for people with RP in terms of how deteriorated their vision is for certain clinical trials? Thank you.
Okay, so first of all, getting back to, there was a question about whether cell therapy would be applicable to USH2A, and the answer is that yes, but remember, it's where you are. It's always better to keep what you've got than replace what you've lost. But if you've gone to the part where there are no cells left, then a gene isn't going to help you because the whole cell is gone. So the answer is yes. So USH2A is part of the trial that we're doing, but you're going to be further along than what would be eligible for those types of trials. Getting back to inclusion and exclusion, it's different for every clinical trial, but essentially all clinical trials are interested in how healthy you are, because nobody wants to, have an experimental drug that everyone's looking at all the potential side effects and have somebody have an untoward effect that's unrelated to the treatment, but still has to be recorded and reported. I'll give you a somewhat kind of off-color example of this, and that was one of the early retinal pigment epithelial trials. They did subretinal injections of retinal pigment epithelium for macular degeneration. One of the patients out of 10, during the course of the year, had rectal prolapse. Right? No one thinks that putting the cells in the person's eye caused that, but it had to be put on the label. Right? Could cause rectal prolapse. Right, exactly. So, I mean, again, so you want to avoid situations like that if you can. So, again, having somebody be healthy enough for the anesthesia, all that sort of thing. So there is no like broad characteristic. But if you are dealing with other health issues that are very significant, then likely no, you're not going to be considered a good candidate. But they are different for different trials.
Okay, thank you so much to our panelists. I think we're going to have to close there. Thank you so much. That was wonderful.